Good afternoon. Welcome and good afternoon, everyone. On behalf of Dr. Sairatushkov, I would like to welcome you all. I will be facilitating today's session. Getinnet is my name, and I'm Assistant Director for Global Research at Penn GHE. This is a Penn GHE's monthly webinar. Our today's speaker is Dr. Chiesa Fuxench. She's an assistant professor at the University of Pennsylvania, Perelman School of Medicine. Dr. Chiesa is a board-certified dermatologist whose clinical work focuses on inflammatory disease with a specific focus in atopic dermatitis and psoriasis. Dr. Chiesa research work is focused on the epidemiology and genetics of inflammatory skin disease, primarily atopic dermatitis, and she's also actively involved in conducting clinical trials in atopic dermatitis. Please join me in welcoming Dr. Chiesa to the floor to give us the speech. Thank you.
Hey. Thank you. Thank you for the very kind introduction and for taking the time to be here this afternoon after a very rainy day this morning. So I do appreciate that. So before we begin, I'd just like to say a few things about myself. I think it's always nice to know who your speaker is and kind of like where they're coming from, aside from what's in the bio. So I'm originally from San Juan, Puerto Rico. That's where I was born and raised. So you see it right there in the map. If you've never seen it before, it's an archipelago consisting of small islands. It's the smallest of the Greater Antilles. To your right, what you're seeing is the School of Medicine where I trained and where I completed my dermatology residency training. And after I finished my training, I joined Penn under the mentorship of one of my current mentors, Dr. Joel Gelfand, which you're seeing a lot of him right here. This is just a picture that his staff did as part of his birthday party celebration where they all dressed up like him. And he has a particular focus in psoriasis, particularly studying psoriasis as a cardiovascular disease risk factor. And he was basically the person that got me really involved in studying inflammatory skin diseases, particularly those chronic inflammatory skin diseases such as psoriasis and atopic dermatitis. And since being here at Penn, I've had the great blessing of being able to work with wonderful colleagues within my department, but also colleagues from different parts of the world, including countries in Latin America, such as my colleagues from Guatemala and Costa Rica. So at least you know there a little bit about me. And so today we're going to talk about a topic that's very interesting. At least I find very interesting, which is trying to understand the genetic basis of some of these chronic inflammatory skin diseases. So when we think about chronic inflammatory skin diseases, we're not just talking about a person that develops a skin abscess, you give them an antibiotic, they're cured, and it's done, right? It doesn't happen again. Chronic inflammatory skin diseases tend to have a couple of things in common. They tend to have a complex pathophysiology. There's just not one single cause. There's multiple genetics and environmental factors that are thought to be at play, and they tend to be chronic. They oftentimes last for a very long time, almost like if you had diabetes or high blood pressure. Like once you get the diagnosis, kind of stays with the patient. And atopic dermatitis, you might know this as atopic eczema or eczema. These are terms that are oftentimes used interchangeably in the literature. Atopic dermatitis is, again, a chronic inflammatory skin disease that is characterized by episodes of acute disease flares. So what do we mean by that? We mean that the patient is otherwise doing fine. They have some sort of baseline level of disease severity, but then all of a sudden something happens. Oftentimes it's very difficult to identify the trigger, and they flare. Their symptoms worsen, right? And they come and they see us. Atopic dermatitis is characterized clinically by the presence of these sort of ill-defined areas of erythema. You're seeing the red patches there. Sometimes those patches are raised and they're called plaques. Oftentimes these plaques, because they can get very infiltrated, you will see areas of increased skin thickening or lichenification. Those are lines that you see across the skin. And it tends to have a predilection for flexural areas. So we're talking about the folds of the skin or the folds behind the legs. Atopic dermatitis is a highly pruritic or itchy skin disease, to the point where oftentimes in clinic, if we have a patient that says that they look like they have eczema, but they say they're not itchy, we sort of take a step back and think about, okay, are we dealing with something else? Because do we have the wrong diagnosis? Atopic dermatitis is also a highly prevalent disease, and this is why I'm particularly interested in studying it. When you think about how common the disease is, it's thought that in the United States, about 10 to 20% of children may be affected by this disease at one point in their lifetime. And when you think about the prevalence in adults, it's estimated to be around 3 to 10% of the population. So we're talking a big group of people here. And also, when you think about atopic dermatitis, oops, how can I get that back? There we go. So atopic dermatitis is not just a skin disease, right? I mentioned previously that patients with atopic dermatitis tend to be particularly itchy, but these are patients who also experience a lot of pain. Their skin is very inflamed, it cracks open. These are patients that oftentimes are not sleeping well. And if you have a child with atopic dermatitis, their parent or caregiver is probably not sleeping as well. These are also patients who will oftentimes miss school because of periods of sickness. They will also, even though they're at work, they're not functioning at their full capacity. And oftentimes they're associated with other comorbidities, such as, for example, higher rates of depression or anxiety and increased suicide risk compared to patients who don't have atopic dermatitis. And they will tend to use more healthcare resources. And if you think about at the societal level, how much does it cost to treat atopic dermatitis in the United States? It's estimated that it's about $5 billion annually in both direct and indirect costs because of the high prevalence of the disease. And so atopic dermatitis does not discriminate between populations, although there have been studies reporting variation in terms of disease prevalence and severity. If you look at the data from the Global Burden of Disease study, again, this was a study that initially was established in 1991 to look at the prevalence, the morbidity, and the number of disability-adjusted life years, or the burden of the disease across populations, including about 200 countries. And one of the diseases that they paid particular attention to was eczema, right? And so what you can see here from the screen is across this closely to 195 countries, you would see that atopic dermatitis is pretty much everywhere across the globe, right? Where you see higher prevalence of atopic dermatitis in countries that are shaded in blue, and then countries that are shaded in green, in orange, yellow, or red as countries that tend to have the highest prevalence of this disease. And in addition to the prevalence of understanding the prevalence of atopic dermatitis, why do we care about this disease, right? And it's the association of atopic dermatitis with other atopic conditions. So if you look at data on comorbidities or diseases that coexist with atopic dermatitis, you start to see sort of a pattern that patients with atopic dermatitis tend to have higher prevalences of, for example, things such as food allergies, asthma, or seasonal allergies, right? And in some cases, it seems to follow a trajectory in what is known as the atopic march. And what the atopic march is saying is it all starts with atopic eczema. And by having atopic eczema, you have a defective skin barrier that allows penetration or transcutaneous penetration of allergens that then result in activation of the immune system. And it's through this mechanism that perhaps these patients go on to develop these other atopic diseases. Now, while this is not true for all patients with atopic dermatitis or all patients with asthma, I think it's an interesting concept because it has us thinking about the potential of, can we prevent this disease from occurring in the first place? And if in doing so, can we modify the occurrence of these other diseases later on or prevent them from forming in the first place? And so when you think about the pathophysiology of atopic dermatitis, as I previously mentioned, there's not just one single cause. When you think about the disease, this is pretty complex. We're thinking that there is an interplay of genetic risk factors, epidermal or skin barrier damage, immune dysregulation, alterations in the skin microbiota, as well as exposure to environmental and other socioeconomic factors. And when we try to explain the pathophysiology of atopic dermatitis, there's different theories, right? There are some people who think that atopic dermatitis is primarily an inside-out disease. It's an immune dysregulation disease where you have allergen presentation and then that results in activation of the Th2 inflammatory cascade that then results in skin barrier dysfunction. And then there are others who are more on this outside-in camp where they say, no, it's not immune dysregulation the problem here. The problem here starts with a skin barrier defect. When you have a skin barrier defect, you have that penetration of allergens that then results in activation of the immune system. I think regardless of which camp you're in, I think both of them are right. I think that you have to think about immune dysregulation. And that is something that a lot of people think about these days. When you go back and you look at the treatment landscape in atopic dermatitis, this is a very busy slide. But what I'm trying to show here, what you're seeing at the bottom of the slide is basically some of the inflammatory signals that have been described in patients with atopic dermatitis. And what you're seeing in the red and blue boxes are the different therapeutic agents that are being developed to tackle this disease. And so most of the agents that are currently being developed are called monoclonal antibodies, and these are targeting specific cytokines that are driving the inflammation. So they're stopping the signal, but we don't really know what sort of this master regulator is, and we don't know what happens in patients where it's mostly the skin barrier defect that's driving the process. And so if we're going to understand a little bit of the work that I want to do, I think it's important to have a basic understanding of the skin, right? So let's focus a little bit on the epidermal barrier. What you're seeing here is, of course, the epidermis. To the bottom of the basement membrane is what you would find the dermis, right? And so the epidermis is composed of a series of layers. You start with your basal cell layer, and you have your keratinocytes, which are the cells of the epidermis. And as these cells continue to differentiate across the different layers of the skin, they change. And at the different stages, you'll start to see different proteins, and those are the proteins that you see listed here on the figure underneath each of these layers. And so we're going to pay particular attention to the granular layer, because it's where you see those little small black dots start to appear, and those are called keratohyalin granules. And within those granules is where you're going to find for the first time a protein called profilagrin. And so as these keratinocytes and as these granules continue to migrate throughout the skin, that protein profilagrin then gets cleaved in a series of steps into a protein called filaggrin, which is important for the pathophysiology of atopic dermatitis. And again, what you're seeing here, if I would just have you focus on that little black square on the top, that is the stratocorneum, and that is where filaggrin actually has more of its important role. So filaggrin, when it's actually secreted, it binds to keratin intermediate filaments, and that's what you're seeing in figure B over to your right-hand side. And by binding these intermediate filaments, what filaggrin is doing is basically providing structural support for those cells on the skin. And then not only that, but filaggrin is also important because as it becomes degraded in the skin, it also results in free amino acids that then form groups. And then the further degradation products of these amino acids go on to become what is known as natural moisturizing factors on the skin. And these natural moisturizing factors are going to be important because they can help regulate the pH of the skin. They can also help regulate transepidermal water loss, so how much water the patient is losing through their skin so they're not drying as much. And they're also thought to have immunomodulatory effects, particularly when it comes to photoprotection. And so what codes profilagrin, like where this is coming from? So profilagrin is encoded primarily by the gene called FLG gene. This gene is found in chromosome 1. It's part of a gene cluster that's known as the epidermal differentiation complex. So there are many other genes that are in there, but it is this particular gene that is composed of 3 different exons that codes for the protein called filaggrin. Typically, the part of the gene that codes primarily for the protein is located in exon 3. And so what you're seeing at the bottom in figure B is what that protein sort of looks like, right? Where you have your N-terminal domain and your C-terminal domain, and then in between, you have about 10 to 12 filaggrin repeats that will vary, right? So not everyone has the same number of repeats. And so what you're seeing here in this figure is that we've known for some time that there are certain variants that are associated with decreased filaggrin expression. So what you're seeing in figure 8 on the top there is perhaps some of the most common variants that have been associated that we've seen also in patients with atopic dermatitis. And what you're seeing here at the bottom of the screen is what is happening when you have those pathogenic variants, right, that result in decreased expression of filaggrin. So in figure B, if you look to the left-hand side, what you're seeing in kind of like the darker brown color, those are the keratohyalin granules. And then what you're seeing at the figure to the right, again, of a skin biopsy, what you're seeing is decreased expression of these granules in patients who have these pathogenic variants. And then what you're seeing in figure C is, again, just an immunoblot showing the decreased production of profilaggrin or filaggrin in patients who, again, have these pathogenic variants. And so, as I previously mentioned, we know that mutations in the filaggrin gene are associated with an increased risk of atopic dermatitis. We know that these patients, not only does it increase your risk for atopic dermatitis, but in some populations, and I do apologize, the text is a bit small here, but in some patients, they might actually predispose to an increased risk of asthma as well, particularly if there is a risk of if the patient also has underlying atopic dermatitis or atopic eczema. And what you're looking at the curves over to your right is, again, that patients who have filaggrin mutations tend to have more persistent disease. It doesn't clear up the same way it would be for patients who don't have these types of mutations. Now, when it was first identified, and this goes back to about 16 years ago, right? When researchers found, it's the filaggrin gene, the FLG gene, this is kind of like the holy grail, and everyone was really excited about that. But the problem with those studies is the lack of populations that are included. So if you go back and you look at these studies, you would see that most of these studies have been done primarily in populations of Northern European descent or Asian populations. And if you look at the percentage of studies that include either African American populations, Black populations, or Latino populations, it is thought that they only comprise about 2% to less than 0.5% respectively of all these studies. So definitely a lack of representation here. And so why is this important? Because of the smaller studies that have been done looking at these other populations, we know that some of these variants are not expressed or are not found across all the populations the same way. So for example, if you focus on the top part of the figure on the top of the screen, you will see in pink the variants that are more commonly identified in patients of European descent, particularly the R501X and the 2282L4. And these variants are known to account for about 90% of the mutational burden in patients with, atopic dermatitis. And then what you're seeing at the bottom of that figure are the variants that have been described primarily in Asian populations. And I would like to point out, for example, 3321-A, that specific variant is identified primarily in Asian populations, but not found in European populations. And then what you're seeing at the bottom of the screen, it's a separate study, but I included here because it did help identify some of the variants that were primarily seen in Black populations or African American populations that were not seen in other groups. And so what about these variants in Latino populations? What do we know? And the problem is that we don't have a lot of data to begin with, right? I was only able to identify it up to this point, one single study that is coming from a group in Santiago, Chile, that included about 500 Chilean patients with atopic dermatitis and their controls. And in this particular study, what they found was that the prevalence of the most common Northern European mutations was similar in this cohort, right? The R501X and the 2282L4. What was interesting about their study was that they really did not find that the presence of these variants, although higher in patients with atopic dermatitis, it didn't seem to correlate with an increased risk for atopic dermatitis. So either maybe this is not important in this population or we're missing other variants. Now, what other data do we have? So I did a small study using data from the peer cohort. So the peer cohort is known as the Pediatric Elective Eczema Registry. And so this is a cohort of children that have been followed prospectively for about 10 years, looking at disease characteristics, looking at disease control throughout their lifetime. And even though it's a very small cohort, I was able to identify a prevalence of any filaggrin variant about 17%, which is higher than what we've seen in other populations. For example, in Northern European populations is about 10%. Interestingly, in this particular cohort, of the four most frequently studied FLG variants, only R501 and S3247 were identified in this cohort. And then what also got me thinking and kind of led to the development of this study that I'm proposing today is the presence of this variant S3316X. So this variant has been previously identified in PEER, but only in patients who are Black or African American. And that got me thinking a little bit about the role of individual ancestries and how it plays into the identification of these variants in patients who are at mix, such as Latino populations. And so when we think about using race as a marker for ancestry, and I'm sure many of you are more of an expert in this area than I am, but we know that race stands for something, right? Race is important, and we should not think about including it in certain areas of research because it can stand for sociodemographic factors that can help us explain certain health disparities. But when you're trying to understand the genetics of a population, it actually may not be a good marker, right? Because when you look at GWAS studies, basically you're grouping people based on broad geographic ancestry, or what is known as global ancestry. And that may hold for certain categories of races. But when you have an mixed population such as Latino, that may not actually hold. And so what you're seeing here at the bottom is a picture of people who are either Puerto Rican and of Puerto Rican descent. And if you were to place them in different racial categories, they would all pretty much fit into different categories just because of what your eyes can see, right? And that's sort of like European bias that we find in these larger GWAS studies. We have to be careful about that because if we use the findings from those studies to develop risk prediction models, right? We might be potentially overestimating or underestimating, disease risk. And why? Because I previously mentioned that individuals who are at mix may have different ancestries at specific regions of the genome that contribute specifically to disease risk. So if you're just looking at someone and just classifying as this person is this, but don't look at those individual ancestry, you may be missing something. And why study Latino populations? Well, I started this talk by saying that I am from Puerto Rico, so I'm biased towards that population, right? But when you think about it, Latinos, Hispanics, however you want to refer to us, are currently the nation's largest ethnic minority. So they're estimated to be about 60 million Latinos in the United States, currently comprising about 14% of the population of the United States. And it's estimated by the year 2060, that amount is going to grow to about 28% of the population, or around 119 million people. It's a big chunk of people that we don't have a lot of data on. And again, when you think about Latinos, it's very hard to categorize them in current racial groups because it's not a race category, right? We can also argue that there's only one human race, right? But when you think about Latinos, there's a pan-ethnic identity, right? So these are mostly countries from Latin America or countries who are of Spanish descent, such as Central, South America, or the Caribbean. And when you think about how Latino populations, how their genomes came to be, it's a very interesting history. And it kind of traces back to three major events that so far have been described that I am familiar with. The first being that green line that you're seeing to kind of like the right-hand side of the figure, I'm sorry, which was the migration of ancestral populations around 15,000 years ago. And then the second large event was the colonization by Europeans, represented in the blue line, which occurred during the 15th and 18th century. And then the African slave trade, the African slave trade coming primarily from West Africa between the 16th and 19th century. And so if you focus on the figure, what you're seeing in the bars are the different chromosomes, and you can see how these different admixture events could potentially lead to what the modern human chromosome would look like today after all these different admixture events. And so again, why am I kind of pressing forward with this idea of looking at ancestry or individual ancestry in Latinos, as well as genetic variation and their association with disease risk? And so there are some studies showing that even within the same group of Latino populations, so for example, if you're only studying, say, Brazilians, right? And some people might say, well, we think they're all the same, right? But even within those particular groups, we start to see differences in terms of the proteins that are being expressed in the skin. And that's what you're seeing here in the blue figure. So you're seeing different groups of Brazilians, and what you're seeing in the smaller darker blue and lighter blue are basically the expression of these skin barrier proteins and also ocular surface proteins. And you're seeing how that can vary depending on the specific quote unquote ancestry of this group. And then what you're seeing over to your right-hand side or left-hand side, I don't know at this point, is basically data from a study that was very interesting. It actually looked at a Puerto Rican population in asthma risk, right? So when you think about who is more prone to develop asthma, there are multiple studies showing that if you're of a lower socioeconomic status, you are at a higher risk of developing asthma. But those findings don't really hold true for Puerto Rican patients or patients that have been studied so far. And some people think that there actually is something else at play that is not only social environmental factors. And so what this study did was basically they recruited I'm confusing the number of patients at this point. I think it's about maybe 300 patients, give or take, but I don't want to be mistaken. And they looked at using ancestry informative markers. They looked at the percentage of individual ancestry. And what they found in this cohort was that, yes, socioeconomic status is important, but it might be that the effect of socioeconomic status actually being modified by the percentage of African ancestry in this cohort. And those patients who had a higher percentage of African ancestry and were of a higher socioeconomic status tended to be at higher increased risk for developing asthma. And so with this sort of kind of story that I've been telling, for the past 30 minutes, that led to the development of this current cohort that we are working on. So if you have any or know of anyone that might be interested, we're more than happy to welcome them into this cohort. It's the Genetics of Atopic Dermatitis in Latino Patients cohort. So this is a cohort where we're trying to recruit patients who self-identify as Latino, but who will have a physician diagnosis of atopic dermatitis because there are issues with how we classify atopic dermatitis. And in an attempt to reduce misclassification, we're trying to have that physician diagnosed confirmation. We'll be collecting data on the history of these patients, not only social demographic factors, but also looking at their parental history and also a generation before that, their grandparents in terms of their ancestry as well. And so where are we recruiting these patients for? So currently our study is open for recruitment. We're looking to recruit patients from the dermatology clinics here at the University of Pennsylvania, but also we're very fortunate to be able to have a collaboration with the University of Puerto Rico School of Medicine, where we hope to kind of identify a more kind of contained population of Puerto Rican patients. We thought about potentially including patients from the Penn Medicine Biobank, but unfortunately, we were only able to identify about 700 unique individuals having an ICD code consistent for atopic dermatitis, and only 13 of those self-identified as Latino. So not a great resource for us at this time. And then our other strategy in terms of recruitment is we're looking to establish community partnerships, particularly with the communities in Northern Philadelphia, right? So if you're in North Philadelphia, it's a large Latino community there. So this is an important group to reach out to see if they would be willing or interested in working with us. And so once we establish this cohort, what is our next step? Because this is a very small grant, which is funded through a K-23 NIH grant, we're only really capable of identifying and describing filaggrin loss of function mutations. Now, there are other genes from that epidermal differentiation complex that I previously mentioned that may be important or could be important, but at this time, we just cannot do that. So what we're doing is basically requesting saliva samples from these patients in addition to filling out our questionnaires. And we are working closely with the Skin Biology Diseases Research Center. So this is within the Department of Dermatology. So if anyone, they have different cores that we can work together. So if anyone's ever interested in establishing some sort of partnership, please reach out to me. I'd be happy to put you in contact with the person. And then we are also partnering with colleagues at the Agency for Science, Technology, and Research. So traditionally, the FOG sequencing has been done using a more traditional Sanger approach to sequencing, which again, you probably are all more experts at this than I am. My understanding of this is that it can be time consuming, especially for this particular gene and labor intensive. So my colleague, Dr. John Common in Singapore, has developed a method where he can sequence this gene and identify not only common variants, but also rare variants that we might not identify with traditional Sanger sequencing. And with that, I would like to thank my mentors and collaborators from the Department of Dermatology, and also Dr. Sarah Tishka for the opportunity to present this work and hear your comments and ideas for improvement. And then I can open the floor to any questions or any comments anyone may have. We have time.
Let's see if colleagues who joined us online may also have.
So I think.
So I have a question when you're, if that's okay.
Yeah.
Go ahead.
Hi, this is Sarah. Great talk. That was really interesting. I guess my first question is, I know that at the filaggrin gene, there are also these repeated, regions, right? So these repetitive elements within the protein or within the gene. How much might that make a difference, like the number of repeats that someone has, as opposed to just like the mutation itself that are within the repeats in terms of risk for the disorder?
I think that's a great question. I think you're, Dr. Tishkov, you're referring to copy number variation.
Yeah.
Right, because based on how this protein is made, those 10 to 12 repeats, so that can vary, right? And so far, work that I've done with Dr. Margolis, which is my mentor for this award, it hasn't really shown, while there might be variation in the copy numbers, it doesn't seem to be associated or affect disease risk. So it may or may not be important at this time, but I think definitely something to continue thinking about, because that's only one study, right? Which again is prone to all kinds of biases, I think. But at this point, at least in the populations that we have studied, we haven't really seen that it affects the risk.
Okay, and then another question I had is, are there any potential like selective advantages that people who have these mutations that put them at risk for the disease, but might they, I don't know, you mentioned something about, I thought was really interesting about the acting as a barrier against maybe even loss of moisture or something like that. Like that was really intriguing in areas that have that are very arid, for example, or in terms of something to do with infectious disease susceptibility. Do you have any thoughts about that?
Yeah, I think that's another great question in terms of why these things are happening, right? And so there is some suggestion in the literature that some of the people that have these types of variants that makes their disease more susceptible to things that are outside in the environment, right? That apparently it was sort of a protective mechanism to develop tolerance to some of these pathogens where, for example, vaccines were not around. So there are people that have suggested that you can see some of these variants in populations that may have been more impacted by the plague as a form of natural vaccination, where they're allowed exposure to these things and they develop tolerance to them, right? Which I think is very interesting to think about, something like that. But yes, that's some of the potential sort of evolutionary advantages that sort of have been described with these types of mutations.
Great, thanks so much.
Thank you.
I have a question too. I probably may have missed it when you were talking, but I wanted to know, what is the prevalence of this condition generally in African ancestry individuals versus Hispanic and then the Caucasian population. I
Yeah.
asked that to ask because on your when you talked about your project that you're planning on doing, you're going to recruit from the hospital, and I'm wondering the prevalence and for the individuals who don't have insurance to be able to be at the clinic to be included in such a study, how that would be, possibly addressed.
Yeah, I think again, that's another important point. When you look at the studies that have examined prevalence in atopic dermatitis, there are a few limitations that I should point out first, right? One of is a misclassification of patients. So oftentimes some of these disease prevalence studies have not often rely on the same definition of their disease. So you could see maybe some variation in the prevalences that have been reported. Now, more recent studies have been trying to do a better job at looking at the sort of standardizing this sort of definition using standardized criteria and applying that across the populations being studied. So far, the studies haven't really shown that there are differences in the prevalence between, for example, Latino populations, but again, Latino groups are usually a minority of the patients that get included in these or have been studied. But they seem to be similar to what we would see in white populations or Black or Asian populations. It seems to be somewhat around the same. Now, that's for overall prevalence. But when you start to look at disease severity and sort of disease persistent, it tends to be more severe in these other groups and can be more persistent. But again, this is based on data that, in my opinion, some of them lack very good study design. So we have to really be careful the sort of kind of conclusions that we make from those studies. But you're right. I think we have to keep in mind, enrolling patients just from our clinics introduces a bias. And that's part of the reason why we think engaging with the community, especially the community in North Philadelphia that may not seek care down here, might be a way for us to get a better sample. It might also be interesting. I mean, the community in North Philadelphia is primarily a Puerto Rican population. So it might be interesting if we have data from that population and then we have the data from Puerto Rico, from Puerto Ricans that currently live in Puerto Rico, could we look at different socioeconomic or environmental factors? But again, that would be, for a larger study that we can't really do that with the amount of funding for this study right now, but definitely questions to look at in the future.
Thank you. Yeah, sounds like it's an interesting
A
question.
similar study like your cohort on African Americans or African Indians going on now.
Right. Not to my knowledge in terms of looking at ancestry. So there are studies looking at FLG variants or mutations, right? But not that it's usually self-reported race, right? So that's what people say, I identify as this and that, and even the peer cohort that I looked into. So peer cohort initially started as a registry for patients with atopic dermatitis, and these were patients that were going to be treated at the time with a medication called the brand name is Elidel, but it's known as pimecrolimus cream. And so the idea was to follow these patients throughout time because there was concern that this topical medication could be associated with increased risk for malignancies and whatnot. And actually, the registry has shown that that is not true. But as part of that registry that looked at African Americans, people were just self-identified by race, so not really looking at ancestry, not really going back and looking at that. So I think for Latinos, I mean, if you ask Latinos how they classify themselves in terms of race, I would say it's very difficult. Like, I wouldn't, even I, who I'm pretty white, have trouble checking the white box because of the concept, right? So doing something like this, when you think about how big this population is going to be in just a couple of years, I think it would be helpful, right? To try to understand. And because there is concern that I've seen studies coming out showing like, well, if you have this variant, it puts you on increased risk, and therefore it should be added to this risk prediction model. And then there's, machine learning that incorporates some of these findings, and now artificial intelligence that will incorporate some of these findings. And I'm like, well, who is the background population that you're getting this data from? That has implications for how you estimate disease risk. And this would be a place to start. So hopefully other people become interested and thinks it's important.
Yeah. Hi, I am interested in the inflammatory role in atopic dermatitis. So what's the role of Th2 cells? I'm not so familiar with
Sure. Oh, sorry. I should have explained that. So when you think about atopic dermatitis, T helper cells. So atopic dermatitis is thought to be primarily as a T helper type 2 disease, right? So when you think about psoriasis, to give you an example, that's primarily considered as a Th1 or Th17 disease. So they produce, for example, IL-17 or tumor necrosis factor. And so those particular cytokines drive the inflammation in psoriasis. And some of the targeted approaches that we have, some of these monoclonal antibodies target those cytokines. In the case of atopic dermatitis, it's primarily thought to be a Th2 driven disease. So for example, interleukin-4, interleukin-13, interleukin-5, those are all classified as Th2 cytokines. They seem to be at opposite ends of the inflammatory spectrum, but I mean, the immune system is not like that. Everything is jumbled together, right? But these patients with atopic dermatitis tend to have more of that inflammatory signal, the Th2. And so that's why if you were to use therapies for for psoriasis, you can't use those same targeted monoclonal antibodies for atopic dermatitis because they don't work. Their problem is not the same signal, right? So you can treat it as much as you want, but it won't respond.
Okay. So the outside trigger factor or the environmental factor trigger atopic dermatitis, are they mainly some microbiomes or?
Yeah, that's a good question. The area of environmental factors, right? When we say environmental factors, that is a whole host of things, right?
Yeah.
We think about there are potential interactions with the skin microbiome and also environmental factors. And actually it's interesting, but we don't know exactly the role. I mean, we know, for example, that if a child is exposed to secondhand smoke or smoking, that increases your risk for atopic dermatitis. There are some studies showing that patients who live in areas that are colder to begin with, that are drier, right, tend to have higher rates of atopic dermatitis. But that's not, I think you have to be careful about that because, and there are studies showing that, for example, populations, for example, there's actually a study that looked at Puerto Ricans who moved from the island to Chicago, and they developed an increased risk of developing eczema. Meaning that they're not in an environment that seems to be more, arid and cold, less humid, and therefore they may have had some genetic susceptibility, and then the environmental factor comes into play, and then, that's the end result. But we don't really know. There's actually studies looking at even exposures in the womb and even colonization with the mother's microbiome at the time of birth as potentially impacting the disease risk. So I do think the microbiome is probably very important.
Yeah.
And also the relationship between, atopic dermatitis and other diseases that are maybe not considered to be of an atopic nature. So for example, we've done studies looking at the risk of developing inflammatory bowel disease, such as Crohn's and ulcerative colitis in patients with atopic dermatitis, and have found an increased risk in patients with atopic dermatitis that increases with disease severity. And we know that there are some genes that seem to be similar in both diseases. So maybe there is some sort of genetic predisposition that we're seeing in these patients. Maybe it's not the disease in and of itself that increases your risk for developing these other diseases. Maybe you are going to have these diseases because that's how you are coded since the beginning, but maybe the presentation of these diseases occurs later on because there needs to be an environmental factor, or maybe we cannot diagnose them before, which is what happens with asthma. Oftentimes, atopic dermatitis gets diagnosed first and then asthma follows, because it's very hard to diagnose asthma early on, right? People tend to kind of move their diagnosis away, but the atopic dermatitis, you see it on the skin, right? You can see if there's a rash there, and then you would call it that. But the asthma, you don't really see it. You can hear, you can listen to the wheezing, right? If you listen to the chest, but it's very interesting, I think, how that would happen.
So the related question is the Th2 cell mainly react to microbiome, or it is reacting to some inside factors?
Right. So what's causing the Th2 to increase the secretion of? So we don't know, right? But we do know that, for example, colonization with certain types of bacteria, such as Staph aureus, can lead to increased expression of IL-4 and IL-13,
Okay, so
right?
That's not the other Th cells, the Th7.
So those would be Th2 cells. Yeah.
Th2,
Right. But it's different to know, like, sort of what came. It's kind of the chicken or the egg, right? Like, what came first? Is it the colonization with these pathogenic bacteria that then leads to activation of these cytokines, or somehow these cytokines were turned on, and then that's leading to impaired skin barrier and allowing colonization of bacteria, which then continues to drive the process. Yeah.
Got a question for Matt?
Thank you.
Matt?
Yeah, a couple questions. One, so I noticed the distribution was also in the eye.
Yeah.
So following on what Sarah was asking about selection, and like, is that the only two places where it's known?
Atopic dermatitis. So, oh, so filaggrin can be expressed, no, so it will express anywhere there's epithelium. So not in mucosal surfaces. So for example, even in the nose, in a certain part of the nose where there's epithelial surfaces, the protein would be expressed there, but not in other parts where it's mostly mucosal tissue.
So the ocular, flagrant ocular tissue. Yeah.
So where there's keratinization. So if there's like keratinocytes, you could have that protein expressed there. Right. Oh, that's an interesting question.
Like, it's not.
Yeah. Yeah, that's a very interesting question, and it might, it just got me thinking about, so, dupilumab is a monoclonal antibody that was developed. It was the first biologic therapy that was developed for patients with atopic dermatitis. It actually came into the market in 2017. So we started being able to use it for patients. And before that, we had no targeted therapies. We only relied on things like prednisone, systemic immunosuppressives that basically shut everything down. We did not know what they're shutting down. Well, in some cases we do, but for the most part, we don't. And so when dupilumab came out in the clinical trials, and we see this actually in clinic, one of the most common side effects that you can see is conjunctivitis, pink eye. So not infectious conjunctivitis. It's mostly thought to be this quote unquote allergic, which no one really knows why it's happening. Some people think it's because you're somehow reducing the expression of goblet cells, and that decreases the production of lubrication in the eye, but we don't really know. Some people think that the eye is kind of like this sequester side, and then now you're messing with all these cytokines, and then you're causing this. Oftentimes patients, fortunately, who develop that side effect can stay on treatment. They don't need to go off of it. You can treat the conjunctivitis with just like lubrication, and that's fine, but sometimes you do need to stop it. But what's interesting about that finding is that it wasn't really seen in patients with asthma or food allergies, where they also do these types of studies. So it seems to be particular for patients with atopic dermatitis. And quite frankly, we don't know why. Like, we're trying to look at it, but part of the issue is that when you had the initial trials, you had dermatologists making the diagnosis of eye disorders, and we're not really good at that. We're just really good at skin.
After the report about this, that's the moisture barrier. And dermatologists measure moisture transfer of the patient's skin?
Yeah, so you can measure transepidermal water loss, and we can do it in clinic. There's a probe that you can put on the skin. You could actually do it in the lab too if you wanted to.
That protein
Yes.
variance.
Yes, you can do that. If you plan on doing that, you have to typically we're doing a clinical trial where we are currently measuring that. We have to keep it in a room where the humidity and the temperature doesn't vary too much. The patient needs to come in and they need to kind of acclimatize, like to just, not be running from the street and get it done because it can alter the results. But you can measure that and you can tie that into, you can look at disease severity, you can look at expression of proteins. It's
Variation.
Mhm. Yeah.
But you don't know, is there much variation?
So we do know, so in terms of whether it changes across population, whether or not people who are in like more northern climates, what we, not so much the geographic variation, more the severity of the disease. So we do know that patients who have more severe disease have greater transepidermal water loss, because you would think, right, that their skin barrier is not as intact and is very compromised. But whether or not I don't think that there are studies that have looked at sort of, people from Northern Europe compared to people from maybe the Caribbean, and looked at how much water, and if it's the same level of disease severity, like if there's maybe like an effect modifier there between location, I don't know. But definitely correlates with disease severity.
Quick question on unlike most inflammatory diseases like asthma or also other autoimmune diseases, which are highly polygenic, like the number of them, you are focusing on one gene, like polygenic. So I just want to hear your reflection on that. That's one. The second one is, what happens during desensitization? When you desensitize from any atopic, and there is a gene already. So I want to understand what's going on during desensitization, and then
So I think atopic dermatitis is different. For example, if you have someone that has an environmental allergy to dogs or cats, or a particular food allergy, or even they have a peanut allergy, right? And you expose them to kind of small amounts of that, can you eventually get them to overcome that allergy, right? And so that's what an allergy would do if they're doing the allergy shots, right? We cannot do the same for atopic dermatitis, because most of the patients that have atopic dermatitis, there is not one thing that is triggering it, that we can desensitize them and say, I wish it were, because it would make my life a whole lot easier, and you could just, maybe cure these patients early on and they can go about their lives and it doesn't impact it that much. But we cannot do that for atopic dermatitis. It's not the same type of sort of immunologically driven disease. And going back to the questions of different genes, I think it's an important question, right? Because when you look at that epidermal differentiation complex that people talk about and throw around, there's many other genes there. There's the FLG2 gene, there's the TSLP gene, and all of these different genes are typically expressed late in the differentiation process of the epidermis, and they code for different proteins that are important for the skin barrier. But so far, and there are studies looking at different genes, but so far the strongest signal across populations has been this one. And so that's part of the reason why I'm interested in, because it's going to be difficult, I think, to study this group from the beginning. And so let's try to go first with what we think we know, and then we can grow from there. But there's definitely room to look at these other genes. There are some studies looking at FLG2 mutations in patients with atopic dermatitis, thinking that these are more common in Blacks or African American patients, but not in white populations.
Yeah, because we know psoriasis, for example, is covered in blood, right? Psoriasis is the other disease that you
Mhm,
is more common, and so I saw in one of your slides
mhm.
asthma, and then anyway, I don't want to take time, but
Sure.
Kathy, you had a question.
I think your disorder is highly heritable,
Mhm.
around like 75%.
Yes.
And then, so I'm curious, like, you had a map with prevalence
But
that, for example, in Africa, it seems like most of the Southern African
Mhm.
countries have like high prevalence.
Mhm.
And then, like, you're saying that there's a lot of, like, obviously people with Hispanic ancestry or, well, from that subgroup, and for example, African Americans have high privilege. But then West Africa, for example, the African ancestry is kind of accomplished. You also mentioned there's like one variant that you think is elevated,
Mhm.
but the odds ratio crosses one in your study, so it might be hard.
Mhm.
So what do you think things like that, like, are causing that? Because also people with like Latin American ancestry also have like the Caribbean ancestry in them, or like an Asian one to some extent, like, well, parts of the parts of Asia, and also I don't think Asia has like something high. So is it like, what do you think would be the reason for that?
So I think,
Right?
Right.
The global burden of disease.
right, so that's the global burden of disease. So again, that's going back to looking at disease prevalence across these different countries, right? So you might see, so it's still prevalent in these populations. I think, again, we have to be careful, like the types of countries that are being included in these studies, right? Like who gets the data recorded and whatnot. In terms of Latino populations, I think what's going to be interesting is if we can eventually start study different groups of Latinos, we might be able to see sort of these different contributions, right? Because not all the stories of Latinos are all different, right? If you look at Latinos from, if we look at a map and we think about even Chile, right? And their study has limitations in the fact that they only study this central population, which is known to carry more of a higher Northern European sort of ancestry. So maybe that's why you're seeing those specific variants there. But if you go to study the more Western populations of Chile, those are people who have more of a Native American kind of influence to them. So what would be the variants that we would find there, right? If you look at Puerto Ricans and you look at sort of the different ancestry contributions, so most of the people from Puerto Rico, it's a higher proportion of European, right? Followed by African American contribution, or Black, we should do it Black. But if you look at Native ancestry, that's not really found in a large percentage of the population because most of the natives were decimated when it was colonized by the Europeans, right? So I think what I would probably start to see, or what I'm expecting to see, is that depending on those different individual ancestry contributions, that somehow is going to correlate with the sort of variance that we're seeing. And that's what I'm ultimately sort of trying to look at.
What's the question?
Yeah, verify that one, and then we got 1 minute, so.
Oh, okay. So quick one. Just first of all, I know that you basically focus on loss of functions. One is the gene expression itself can relate to the AD disease.
It could, yes.
Yeah, because it's a huge problem, right?
Mhm.
It's like almost 5,000 acids. So I mean, just wondering, because based on the slide you show, they have the stop coding everywhere.
Mhm.
I'm just wondering, the different size of the truncations are all related to the.
It can,
Lines.
Right. So there are some studies showing that even if you have a truncated protein, that protein may still be functional, right? So yes, there might be something there, and whether or not that relates to the copy numbers, we don't know. So, for example, there is some data to suggest that you don't need a lot of these like only if you have a few, like a short protein, if it's like a certain amount of amino acid residues, that it could still be a functioning protein, even though it's not the full protein. So yes, that might be something that might be worthwhile looking at in the future. With the current resources that I have for this study, we're not able to look at that, but I think that would be sort of kind of a next step or something to continue to look into.
Yeah, so with that, we end our today's session, and thanks very much, Dr. Chiesa.
And thank you for your time.
Thanks for joining us online and also here face to face. Yeah, wish you all a very happy holidays and have a good one. Yeah.
Thank you for all the great questions. I appreciate it.
Thank you. Fantastic talk.
Thank you. Happy holidays, everyone.
Happy holiday.